Dear colleagues and friends, I am Raffaella Greco, an hematologist in the bone marrow transplantation unit of the Raffaele Hospital in Milan, in Italy, but I'm currently chairing also the EBMT autoimmune disease working party, and this is the reason why today I have been asked to give you some highlights on potential new CAR T-cell indication from the EBMT-EHA CAR T meeting we have seen in the few days before. The entire meeting was really exciting, and you have seen all the latest clinical results of CAR T-cell therapy in its main indication, but also some future indication and new standard of care in this field. And I have had particularly the honor to chair, together with Professor Michael Hudacek, the great session on new CAR T-cell indication on February 11. And undoubtedly, CAR T-cell represent a turning point in cancer immunotherapy, and the great potential to modulate cell activation and redirect antigen specificity made by this CAR T-cell approach, attractive also in new areas such as autoimmunity, infection, and also inflammation, as we have seen in this fascinating session. We have seen really 4 outstanding presentations in this session 6. One from Professor Jonathan Epstein from Pennsylvania, United States. He presented CAR T-cell application on cardiac fibrosis observed in nearly every form of myocardial disease, and he demonstrated the efficacy of redirecting T-cell immunotherapy to specifically target pathological cardiac fibrosis from cardiac fibroblasts in the heart. Adoptive transfer of T-cells that express a chimeric antigen receptor against fibroblast activation protein results in a significant reduction in cardiac fibrosis and restoration of function after injury in animal models. This provides really proof of principle for the development of these immunotherapeutic drugs for the treatment of cardiac disease, for example. Importantly, also, Professor Andreas Mackensen from Erlangen, Germany, presented the first data available on the use of CAR T-cell therapy to treat patients with systemic lupus. The immunotherapy approach induced rapid clinical remission of severe and refractory disease with no notably adverse effect up to now. Autoreactive B cells, as you know, have been long a target for lupus patients, but the efficacy of existing B cell depleting drugs has been unsatisfactory up to now. Conversely, CAR T-cell approach, genetically engineered to recognize CD19, may achieve more complete B cell depletion in this setting. So clinical remission was accompanied by sustained depletion of circulated B cells in this patient, and the rapid disappearance of antinuclear DNA antibodies is also important in this setting. Data on long-term follow-up of these patients are needed to properly check whether these B cells will return, and with them eventually autoimmunity without further treatment after CAR T-cell. But the demonstration of the safety and feasibility of this treatment is really encouraging, as we have seen. And CAR T-cell treatment may indeed a really breakthrough into a new era of immunotherapy also for rheumatic disorders. Moreover, we have seen the presentation from Professor Ulrike Protzer from Munich in Germany, and she has focused on T-cell therapy with T-cell redirected against hepatitis B virus infected cells by virus-specific receptor. This is a promising therapeutic approach for the treatment potentially of chronic hepatitis B and hepatitis B-associated cancer also. She proposed a safety switch mechanism like Cas9 or herpes simplex virus thymidine kinase, this to limit cytokine release syndrome and hepatotoxicity that potentially can occur due to the high number of target cells in this setting. Overall, T cells co-expressing Cas9 and HBV-specific receptor efficiently recognize and kill HBV replicating cells containing an unwanted hepatotoxicity in this peculiar application. Finally, we have seen Professor David Scott from Bethesda, United States, and he mentioned the clinical trials with expanded Tregs in the setting of autoimmunity initiated over a decade ago in transplantation and autoimmunity field. While these cells have been used safely in multiple clinical trials, they are polyclonal and the frequency of specific Tregs is very low. Expanded Tregs express a broad repertoire of specificity and have the potential to be non-specifically immunosuppressive. To overcome this issue and their rare frequency, his lab has focused on the use of specifically engineered Tregs to suppress adverse immune responses in immunogenic disorder, but also autoimmunity and also allergy. He will clarify advantages and disadvantages disadvantages of the different Treg approaches, including TCR, CAR, and BAR, demonstrating how Tregs can be made specific to treat a variety of adverse immune responses. So the choice of which specific receptor to employ mostly will depend on the targeted disease we will have. So we are really approaching overall the future with all these promising and innovative results, paving the way for future study to further investigate this innovative use of CAR T-cell therapy to control autoimmunity and infection. And this is bringing a lot of hope for our patients and also their families, I hope.